Hello and welcome to this presentation on the gut microbiota and its link to health. My name is Jordan and I'm a clinical physiologist here at the Functional Gut Clinic. In this talk, we will distinguish the difference between the microbiota and the microbiome, and another term that you may or may not have come across before, dysbiosis. We will explore the gut-brain axis and highlight some interesting scientific research that shows just how bacteria can influence our brain health and how we feel emotionally. Then we will look at how the microbiota influence other health conditions such as our weight and metabolism. So there are more bacterial cells in our gut than there are human cells in our entire body. In the average human, there are around 30 trillion human cells, but 39 trillion bacterial cells in the gut. We often call this collection of bacteria the microbiota or the microbiome. Now, these two terms are used interchangeably, but the microbiome refers to the collection of genes or genetic material from all the microorganisms within an environment. And it's not just bacteria. There are also fungi, archaea, protozoa, viruses, whereas the microbiota relates to specific names or types of microorganisms within an environment. And you may also hear the term microbiota, which refers to fungi and yeast specifically. We most commonly talk about the gut microbiome, but there are microbiomes everywhere, on your skin, on your hair, in your mouth, and if you're a female, a vaginal microbiome, which is very important for reproductive health and the health of your babies. So several studies have shown that babies born through C-section, i.e. those who aren't exposed to their mother's vaginal microbiome, are more likely to experience food allergies as they get older. Babies are thought to come out sterile, so it's important to get that initial exposure in order to develop their own microbiota. For example, you can see in the figure that the vaginal microbiome is typically rich in the genus Firmicutes, and then in the colon, these Firmicutes go on to make up the majority of the microbiome in healthy adults. But children born through C-section often have a deficiency of these bacteria in their gut and increased allergies to foods like milk and skin allergies like eczema. This shows just how critical the gut bacteria are for the development of our immune systems. In fact, the gut holds around 70% of the body's immune system, and we need there to be a good balance of microbes in order for the immune system to work well. In the gut, we have different types of bacteria. The symbionts, which give us health benefits, the commensals, which don't really provide us with any benefit, and the pathogens, which like to cause disease. Dysbiosis occurs when there is an imbalance in these bacteria, typically too many pathogens. There are many causes of dysbiosis: a poor or restrictive diet, previous gut infection or food poisoning, and medications such as antibiotics which wipe out the gut flora, or things like PPIs, omeprazole, Nexium, which reduce the stomach acid. Many studies have shown a lack of diversity or disruption to the gut microbiome, especially early in life, results in chronic illness like autoimmune disease, IBS, inflammatory bowel disease, obesity, diabetes, and now disorders associated with the brain like Alzheimer's, autism, and even anxiety. So these findings appear to be related to something called the gut-brain axis. There is a special relationship between the gut and the brain called the gut-brain axis. It's sort of a bi-directional link between the central nervous system, the CNS, and the enteric nervous system, the ENS. So we call the gut our second brain because the gut is controlled by the enteric nervous system, which works separately from the brain from the central nervous system. This includes a direct relationship between the cognitive and the emotional centers in our brain. For example, 95% of the body's serotonin is made in the gut by enterochromaffin cells. Serotonin is a neurotransmitter in the brain which makes us feel happy. But in the gut, serotonin has several actions. It controls secretions, it controls motility, so how fast things move through the bowel, and it controls pain perception, so how sensitive you are to pain. Serotonin plays a major role in IBS, Those with constipation often have lower levels of serotonin. The muscles in the rectums are less reactive to serotonin, and they're more likely to have hard or lumpy stools. And then those with IBS and high levels of serotonin can have diarrhea, and the rectums are more reactive with loose or watery stools. So this can also lead to fibromyalgia, which is a heightened sensitivity to pain in muscles throughout the body. And because of serotonin's effects on mood in the brain, many patients with these conditions also suffer from depression. But what is really fascinating is the gut microbiota significantly influence our serotonin production. The gut microbiota plays an important role in the metabolism of the amino acid tryptophan, and this is the precursor required for the synthesis of serotonin in the gut. But tryptophan may also be converted to kynurenine by certain bacteria. In contrast to serotonin, increased kynurenine is associated with anxiety. So, studies have shown that mice receiving the gut microbiota from depressed patients exhibit increased anxiety-like behaviours, and this is in line with higher kynurenine levels. Inflammation appears to shift tryptophan metabolism from serotonin to kynurenine, but anti-inflammatory short-chain fatty acids, such as butyrate, can actually prevent this, and certain species in the gut produce these short-chain fatty acids. For example, species on the left, like Bifidobacterium and Lactobacillus, can be protective against the development of anxiety behaviours by increasing serotonin and decreasing kynurenine levels. Tryptophan is found sparingly in plant foods like oats and beans, but it's highest in meat. A recent epidemiological study showed that people who don't consume red meat are more likely to suffer from depression. This is interesting, and it highlights how diet and bacteria can significantly impact our emotional well-being. The gut bacteria may also have a more detrimental effect on our cognitive health. The gut-brain or gut microbiota brain axis has now been well studied in Alzheimer's disease, and it shows that Alzheimer's may be associated with inflammation caused by an imbalance in the gut microbiota. So in the intestines of Alzheimer's patients, there are higher levels of pro-inflammatory pathogenic species like Escherichia and Shigella, but a decreased abundance of anti-inflammatory species like Eubacterium. This dysbiosis causes gut inflammation and something called increased intestinal hyperpermeability, or leaky gut. Bacteria, they produce these toxins called lipopolysaccharides, and these can leak through the gut lining and cause chronic inflammation throughout the body and even in the brain. In fact, one study found the presence of bacterial lipopolysaccharide was 21-fold higher in the hippocampus, so the part of the brain responsible for memory, of Alzheimer's patients. Lipopolysaccharide toxins are produced by gram-negative bacteria. The most common example is an endotoxin called cytolethal distendin toxin, or CDTB, and this is produced by bacteria that cause food poisoning, such as E. coli, Salmonella, and C. diff. This toxin is the primary cause of post-infectious IBS and leaky gut. Now, leaky gut used to be a bit of a scapegoat for unexplained GI issues, but science is now understanding intestinal hyperpermeability may be associated with many other diseases. If you take a look at the diagram, you can see our intestinal lining is only one cell thick, and this is to allow the absorption of nutrients, but it must also prevent the gut microbes and toxins from entering the rest of our body. So we have this physical mucus barrier, and below an immune barrier in the lamina propria. And this is where the immune system cells constantly monitor the gut microbiome and modulate the immune system. In between our intestinal epithelial cells, we have these tight junction proteins, and you can sort of think of them like security at the front of a concert, preventing the screaming fans from getting on the stage or the gut bacteria and toxins from entering the body. However, these bacterial lipopolysaccharides are detected by our immune cells, and this causes an inflammatory response, and then we get a loss of the tight junction proteins. So without the security, the bacteria and toxins are free to slip through the GI lining and into the body, causing chronic inflammation. And this is what we call intestinal hyperpermeability or leaky gut. The gut microbiota can also influence our weight and metabolism. Scientists first assumed the bacteria in our guts weigh around 2 kilograms collectively. That's the equivalent of 2 bags of sugar. But new research suggests it's not that much, more like 200 grams or the same as a grapefruit. Bacteria affect our weight in a different way, and it has something to do with those short-chain fatty acids. When bacteria break down carbohydrates from our diet, they produce gases and metabolic byproducts, which include these short-chain fatty acids. So we spoke about butyrate already, but there is also acetate and propionate, and these short-chain fatty acids account for around 10% of our daily calories, and they even influence blood sugar control. For example, propionate is associated with increased insulin sensitivity, whereas acetate is negatively associated with insulin sensitivity. There are also certain types of microbes in the gut associated with obesity. There is a Bacteroidetes and Firmicutes ratio, where lean people tend to have a higher abundance of Bacteroidetes and Firmicutes tends to be higher in obesity. Studies by a group in the States also found obese individuals had higher levels of microbes called methanogens, and these produce methane gas. Methane is something we associate with cows and global warming, but around 30 to 50% of us have these bugs in our gut. The main species is Methanobrevibacter smithii, and studies have shown that patients who have bariatric surgery, things like gastric bands, go on to experience less weight loss after surgery if they have an overgrowth of methanogens in their gut. So it's thought that methanogens can actually enhance the production of short-chain fatty acids by bacteria, and this is probably because methanogens also use things like acetate for methane production. So, people from sub-Saharan Africa were found to have higher concentrations of methanogens. It suggests they've adapted to live with these methanogens in their guts, which allows them to extract more calories from low-calorie, non-digestible foods. And if these people were to eat a Western diet, they would likely put on weight very quickly. Intestinal methanogen overgrowth is also a sign of dysbiosis. It's been associated with excessive wind like bloating, belching and flatulence, and also associated with constipation, as methane appears to slow intestinal motility. Now, methane can be assessed through hydrogen and methane breath testing. It's a simple and non-invasive way of detecting the level of methane in the guts of patients. So once methane is produced, it crosses the gut lining into the blood, where it's transported to the lungs and excreted in the breath. Patients with IBS-C, so constipation predominant IBS, have high levels of breath methane. At the Functional Gut Clinic, we're also doing some groundbreaking research into how iron supplements may cause constipation by actually enhancing methane production in the gut. So, as we come to the end of this presentation, we will just summarise what was discussed. Microbial diversity is good, especially in early life to promote immune system development. Dysbiosis is an imbalance in symbiotic and pathogenic bacteria, often caused by disruption to the gut microbiota through antibiotics or things like gut infections. We learned that the gut microbiota brain axis controls our mood through tryptophan metabolism, and leaky gut is actually an emerging problem in many health conditions. Lastly, we learned that certain microorganisms like methanogens influence our weight and metabolism, and it's quite simple to test for these through hydrogen and methane breath testing.